right, good morning, everyone. Okay. So before we get started today, I wanted to take a few seconds and talk about some sort of housekeeping stuff. The first is that you may have noticed that the questions are a little bit wonky for quiz 6. It looks at first glance like I think I mixed up 6 and 8 somehow when I was putting stuff together. So if you've sort of looked at chapter 6 and panicked, that might just be that I've pulled the questions from the wrong place. So I'm going to fix that. And then what I'll do, because it's so close to the deadline for it, I'm just going to give you guys a couple extra days for it. Probably just for ease, I'll bump the due date by a week so that you have access to it for a while. But I'm going to try and fix it today. I just haven't had a chance to sit down and figure out where have I pulled these from and why are they not lining up with what we were talking about. So I think it's that I mixed up 6 and 8, but we'll see what happens there. So I'll just make a note on Moodle for you whenever I figure out what's going on there. I also wanted to ask, have we actually gone over the written assignment in the class? I have a very similar assignment for a couple of different classes, and I can't remember if we actually sat down and walked through what's on Moodle. I'm getting some blank looks, so apparently. Okay. Okay, then I will refresh it now. Yeah, clearly my memory's not the greatest, so I was having a moment of, did we actually do this? We should probably do this. So we'll start just by running through that. So with the written assignment, if you've looked at it, you might have realized that I tend to be a lot more wordy in my explanations. I do it in person as well, but when it's writing, I like to give you guys sort of all the extra information possible. I also tend to enjoy preemptively answering questions because I use fairly similar assignments between different classes, and I know what tends to trip students up. So my descriptions seem to get longer and longer each year. But if there is information that's missing, you can always let me know, and I'll sort of improve for the future. But the short version of what's here on our web page is that I'm trying to get you to look critically at some scientific media. So I want you to compare a popular media article to the science that it's based on. So the first step here would be to find a popular media article. How I typically do this, so whenever students are stuck and they're not quite sure, I usually go into Google and search for phrases that appear in a lot of media articles. So I'd say scientists have found that, or researchers show that. And that'll usually get you to a pretty good place. And then if it's a good media article, they should link you to the original research. So they would send you to the paper that they're talking about or the original study that they're talking about. And so I just want you to do some critical thinking and say, okay, how does the popular media article compare to the scientific media or scientific journal article? And so the idea here is if it's a good popular media article, then it should be the same information as in the journal article, but maybe presented a little bit more clearly with less jargon and scientific terminology so that it's accessible for the general public. If it's a bad popular media article, then it might take some liberties with what the study actually found. And so we typically see they tend to be on the bad side, at least from my experience reading student submissions, where they'll sort of skip out on important details. And so I'm getting you guys to look at them and say, are they making a claim that maybe isn't backed up by the research? Or have they completely skipped over something that would really help us put this information in context? And so I have 3 simple questions that are getting you to sort of start thinking critically about this. The first is, what does the popular media article claim? What is it that they're saying the research has found? So a simple one sentence, what are they claiming? What are they saying has been found in the research? Then we're going to do the critical analysis part. So how confident are you in the claims? Do you think that the article's doing a good job, or have they taken liberties? What kind of information are they missing? And for this one, I've said specifically, show some critical thinking. Do you have personal experience that you think that it's doing a good job or making a good point? Or do you think that it's sort of missing out on something important that you think should be mentioned? So I'm not looking for a specific exact right answer here. I'm just looking at you to employ some critical thinking. And then our third question is, okay, now that you've looked at just the popular media article and sort of critically evaluated its content, how does it compare back to the scientific article? Go back to the original journal, look at the study. And here I specify you only have to read the abstract because some of these papers can be massive. I have some in my collection that are 30, 40, 50 plus pages long. There is no need to read any of that. Just focus on the abstract, so a nice concise summary, and the authors should give you a sort of, here's the important points to take away from this. So looking at that, can you make any conclusions about the differences between the media article and the journal article? And it's not to say that, like, if you wanted to go and get more information from the article proper, you can read into it. I'm just saying you don't have to. And then the last one here is just to include a proper APA citation, like a reference for each of your two sources. So you don't have to be doing in-text citations for the questions, because for the assignment, I've asked you to look at two sources and compare them. So I'm going to assume your information is coming from your two sources. So we're just covering our bases by having you do a citation at the end. So it doesn't need a proper reference page. You can just put the number 4 on your document and fill in your two references. Just watch that. So a popular media article might just be like a website, it might be a blog, it might be sort of any kind of source. So just double check the best way to cite that kind of a source, because it won't be the same as a journal article. And I do find that that trips up some people. But yeah, those are our main points here. And from sort of the way that I mark things is that if something's worth like 1 point, I'm looking for you to make one argument or a statement. So the question that's worth 3 points, if you made 3 distinct statements, that would fulfill the requirements for that question. So you could figure it out pretty quickly that this isn't intended to be a massively long assignment. I've seen people get full marks with as few as 6 sentences. So you don't have to write like paragraphs and paragraphs. This should theoretically fit on a single page. I won't penalize you for going over, but usually in scientific writing, less is going to be more. So focus on just being concise and sort of saying what you need to say. And if you can get it done early, if you wanted to ask questions about something, maybe you're not sure if you've said it succinctly enough or something like that, you can always send your document to me in advance and just say, hey, can you double check this thing? Or could you take a look at that thing? Just make sure that you give me lots of time. And then for submitting it, we're going to get you to save it as a PDF. That way formatting doesn't get wonky. My only requirements for formatting is that you number them clearly. So questions 1 to 4, have 1 through 4 somewhere on your document so I can tell what you're answering where. But you don't have to do, like, no need for a title page or any of the other APA formatting things. The only thing in APA is your references. But yeah, I think that was the important stuff for that. So either it is new information that we haven't done before, or it's a refresher, but I just figured that especially with us coming up on the midterm and then reading week, we should probably go over it again. So you can work on that now, or you can work on it over reading week, whatever works for you, but it's going to be due on November 16th. And I have it set up to be due at basically as close to midnight as you can get using Moodle. So 11:59 p.m. I typically don't mind if you're a few minutes over a deadline. I know that stuff happens with uploads. I know, especially last night myself, I lost my internet for about 4 hours last night for just no reason, which interfered with a lot of my slides and work and stuff. So things happen. But just let me know if anything weird happens. But if you're like a minute over the deadline, I'm not gonna penalize you for that. But yeah, I think that was everything I wanted to say there. Okay. And yeah, the submission portal is there. So if you're done and you want it out of your hands, you can submit it whenever you'd like. And then, yeah, I'll start grading them once everything's submitted. Okay. Alrighty, so now we can actually move on to chapter 8. And I had a little bit of a moment this morning. I saved everything before I left, and then my internet dropped out again. So I had a moment of sort of heart attack here where I logged in and it wasn't showing my slides. But they came in eventually, so I think it reconnected at home, but I just didn't rehearse as much as I might have liked. So we'll see how much we can get done today. And this chapter is going to be fairly similar to what we were looking at in the last chapter, where there's quite a bit of information on different brain areas and eventually some different pathways. But I've tried to sort of break it down a little bit more just to make it easier to understand. And the standard, this is not going to be on the midterm. Everything on the midterm was already covered, so we're only doing up to chapter 6. Chapter 8 onward is what's going to be on the final exam. So you don't have to worry about learning all of this in time for Tuesday. Okay, so all of that out of the way, we're going to start talking about emotion and health. And the way that this chapter is broken up, the first part of the chapter is dealing with emotion and what we know about emotion and a little bit about how we know that. And then we break into sort of how health is impacted by that and looking at stress and coping and other things like that. So today, I think we're just going to focus on the emotion side of things, and then after the midterm, we'll go into the health and stress side of things as well. But we start off with our usual definition. So if we're looking at emotion, how do we define emotion? And the textbook does a great job explaining that it's really, really hard to put a solid definition on emotion. And they even go so far as to say that we all know what emotion is until we have to put it into words. So our very wordy and sort of lengthy definition is that emotion would be an increase or a decrease in physiological activity accompanied by the feelings that are characteristic of that emotion. And this would often also be accompanied by characteristic behaviors and or facial expressions. So all of that is basically saying that emotion is the feelings that we have, the sort of characteristics that we also display while having those feelings, and our interpretation thereof. So not necessarily the clearest of definitions. And we kind of accept that because we're into these more psychology side of things, definitions, concepts, where stuff isn't necessarily as concrete as we might like them. So what you might find with a lot of emotion research is in their literature searches, they'll give you what definition of emotion fits with their work. So there's lots of different definitions based on what you focus in on. Some people may be focused on feelings. Some might be looking at just brain activation. Some might be looking at a combination of both. So when you're looking into it in more depth, you're going to start seeing, okay, well, what do they care about? What aspect of this sort of three-pronged definition do they focus on? So we're keeping it broad and we're saying that it's sort of the activities, the feelings, and then the actions that go along with it. And we end up doing quite a bit of review here because we're going to start talking about different parts of the brain that we've already mentioned. So it's kind of nice. We have that solid background built up from previous chapters, and now we can just add to it. So if we go back to our knowledge of our sympathetic nervous system, and we know that our sympathetic and parasympathetic nervous system kind of work in conjunction, one turning things on and one turning things off, more or less. But our sympathetic nervous system is the one that's responsible for activating our body during arousal. And when we're talking about emotion, we're just really talking about sort of heightened sense of emotion or heightened bodily activity. That's what we're referring to for arousal here. And if we need to be more specific, we can specify what kind of arousal. But for in general, we'll see an increase in heart rate, respiration, our sweat glands will kick into action, all of those things that are characteristic of our fight or flight response. It also shuts down digestion. So even though we think of sympathetic as being an activating system, we do have that sort of, there are some things that get shut down, but it makes sense that they're being shut down. If you're in a fight or flight situation, you don't want to be wasting energy digesting food, because food is something that will be useful in the future, but at this very moment, your energy would be better spent going to your muscles or your brain to help you survive the current encounter. So that's why some parts of the body get shut down. We also have some constriction in our peripheral blood vessels. So the blood vessels sort of away from our core. And so that helps raise our blood pressure and keeps blood flowing to our muscles and internal organs. So again, sort of shutting down certain things to prioritize what's important in a fight or flight moment. And then we'll also start talking about some of the hormones that are involved in some of these responses, especially when it comes to stress. So we'll talk quite a lot about cortisol in the future, but our sympathetic nervous system ties directly to our adrenal glands, which would then emit or secrete, I guess, cortisol and other hormones that can help control these systems. So that's the activation. What about our parasympathetic nervous system? So when our parasympathetic nervous system activates, this is usually shutting things down or getting us back to normal after being in a state of arousal. So after our sympathetic nervous system has done its job, the parasympathetic nervous system comes back on and gets us back to our normal baselines. So we're going to decrease our heart rate and our respiration, get those back to normal. We're going to resume digestion. We're going to redilate our blood vessels. Everything's going to return to our sort of general functioning state. And you'll usually see diagrams that have sort of our sympathetic and parasympathetic nervous systems as opposites for different activities. And they do operate sort of in direct contrast to one another for most systems in the body. All right, so that's our sympathetic and parasympathetic nervous system, but it's not just nervous system stuff that's involved in feelings of emotion or general arousal, as we're calling it right now. We also have the aspect of muscular activation. So basically, what are our muscles doing? Is our body tense? Is our face smiling? These are all sort of different instances of muscle activation. And these activations are involved in our external expression of emotion. So these would be those actions that are stereotypical for different feelings or emotions. So we can have things like smiling and frowning to show that we're happy or sad, but we can also have our body responding, say, fearfully to something. Maybe you run away from a scary stimulus, or maybe you flinch at a loud sound. That's also muscular activation because our muscles are doing things that's sort of lining up with what we're feeling at the moment. And so this combination of our nervous system and our muscular arousal sort of working together, this ends up being adaptive. And it makes sense why these systems exist, because they help prepare our body for an emergency. We tense up when we're stressed because we're waiting to have to react to something. So if we were in the wild evading a predator, being tense would allow us to react more quickly, to duck out of the way, avoid getting injured, or also just being on higher alert so that we're aware of incoming threats. So this is helpful because we're ready for emergency situations. We can act out that appropriate response in a lot more or in a more timely manner than we would if we weren't prepared, if we weren't ready for it. There's also some discussion of this muscular activation actually playing a role in emotion. So there's a lot of debate here, and we're going to get into a little bit of the discussion on what role do our sort of physical activities, do our things like smiling and frowning, how do they affect the emotions that we feel? Are they a cause? Are they an effect? Are they somewhere in the middle? And it's going to depend on which perspective sort of most appeals to you or most fits with the research you're looking at. And so this is going to be another one of those cases where there isn't necessarily a right or wrong theory. They both have merits and support, but neither of them tells the whole picture. So just keep that in mind as we're talking about these two major theories. The first that you might have heard of back in 104 is the James Lange theory. And this basically states that emotional experience comes from physiological arousal that comes before it. So our body reacts to things. We have different patterns of arousal, and that leads to us experiencing emotion. And so the quote at the bottom says, "We feel sorry because we cry, angry because we strike, and afraid because we tremble." So we have to sort of fit what we're feeling based on what our body is experiencing. So it's almost like your body reacts, and then you figure out what emotion fits with that reaction. And we have some early support for this theory because we have different patterns of physiological arousal, especially in the brain, accompanying different emotions. So we have, if we look at, say, a brain scan, we can see activity in different parts of the brain, whether you're happy or sad. So we can actually pinpoint, there are distinct levels of activation depending on which emotion you're reporting. But that doesn't necessarily tell us which came first. Are we having those patterns leading to emotion, or is the emotion leading to the patterns? It's harder to tell. And so our next theory is the cognitive theory. And this is the belief that the identity of an emotion is based on cognitive assessment of a situation. So our physiological arousal is only going to tell us about the emotion's intensity. So when we decide what emotion we're feeling, we're going to decide that by looking at a situation, thinking about it, and evaluating what is our most likely sort of reaction. So this would explain how we misidentify emotions. So maybe you're in a situation where everything is really intense, and you're thinking, well, maybe it's because I'm excited for something. But in fact, maybe it was more nervousness, but you misidentified it because it has very similar sort of traits to it. And then the amount of arousal in your system would tell you if you are sort of very emotional or not very emotional. And they point to research demonstrating sort of mix-ups in identity of emotions, where they had sort of young men who were interviewed by an attractive young woman while they were doing something dangerous, or say 10 minutes after they did something dangerous. And in this case, they were walking on a really high bridge that looked scary. And so that sort of nervous system and bodily arousal was misattributed to feeling attracted to the woman, because they found that those who were interviewed while their heart rate was up from being in a scary situation, they reported being a lot more attracted to that individual, and they were a lot more likely to actually call her if she provided her phone number. And the people who were interviewed after the fact, sort of away from that high energy situation, they were a lot less likely to sort of misattribute that feeling to arousal, and specifically sexual arousal here. So that kind of explains why we might misidentify things if we're not certain what's pushing that to become. So if we're trying to assign sort of meaning to something based on what we evaluate, our evaluations could be flawed, and then we determine it to be the wrong emotion. Okay. And so this is where that which one is correct comes in, and neither of them does a perfect job of explaining this. And if you continue on in emotional research, you'll find that these are sort of the two most popular theories, but there are dozens of different theories of emotion. And some of them are fairly far-fetched, some of them have lots of support, but it seems very difficult to get any kind of consensus on what's actually happening. So both of these have some evidence and some support, but neither is the whole story. And so we see other factors that kind of contradict these and tell us that there's more to it. So we have some studies where if you manipulate a person's facial expression, you can actually change how they feel and how they interpret things from their environment. So you've probably seen or at least heard about some of those studies where they get people to hold a pen or a pencil between their teeth, and doing that action forces a smile. If people are then asked to evaluate different images or sounds, they're going to be a lot more likely to say that they are happy or at least positive than people who hold the pen between their lips, which would be more of a frowning expression. And just changing facial expressions can completely change their interpretation of stimuli. So something can go from neutral to positive or negative depending on what your face is doing. And there are some really cool studies mentioned in the textbook looking at things like individuals who've had Botox done, which paralyzes some of your face muscles. If you're unable to, say, frown because of Botox, you are less likely to report things being negative. And we even see sort of reduced brain activation associated with this. So if someone can't make a sad face, then they're going to show less activation in the part of their brain related to that emotion, which is kind of interesting. So that's neither of the theories exclusively, but it's something that seems to be pretty universal in lots and lots of different experiments. And there is some idea that this, having these facial expressions and having different bodily reactions associated with emotions is also involved in empathy. So when we're talking to someone, you might not realize that you do this, but we automatically start mimicking the person that we're interacting with. It's sort of human nature to copy them in order to either sort of connect with them or whatever. It's something that you're not necessarily aware of doing. But you typically adopt a little bit of their their mannerisms, which you might have found if you spend a lot of time with the same people. Maybe you find that you sound a little bit like them after a while, or you talk the same way. Oh, yep.
I heard something kind of interesting that they looked at like a bunch of like old married couples, and they noticed that they looked more similar than they did when they were young because they have the same mannerisms and their facial expressions, so they wrinkle kind of the same way. It's kind of cool.
It really is. And it's some of the research too has looked at, so there are some people who have, say, maybe an inability to recognize those emotions or an inability to mimic them, and it can interfere with their ability to feel empathy. Because I think the text at least talks about individuals with autism where they have a delayed reaction to that, so they don't mimic as quickly as non-autistic individuals. So they have sort of issues with empathizing and understanding what's going on, at least in some studies. And it seems to be one of those sort of newer areas of research because they don't have tons of concrete information on it yet. But it's neat that they're starting to look at stuff like this to see, okay, well, why do some people have trouble connecting with others? And it's weird. They can look at correlations of the degree to which you imitate someone, and people who have high levels of empathy will be more similar to the people that they're interacting with. So they'll adopt speech mannerisms and behavior mannerisms. They'll adopt postures and expressions, sometimes even at a level that we can't perceive using our own sort of visual systems. They'd have to use a computer program to pick up on micro expressions. But people who do more mimicking tend to also score higher in empathy. So this copying helps us feel the same emotions and then also connect more deeply with others who are feeling those emotions. So lots of really, really interesting research in this area. Okay. And then if we wanted to look at sort of our physiological responses, we actually have parts of our brain that light up when we see people doing an action, but also when we do that same action. So these are called mirror neurons. So if we're focusing in on sort of specific neurons or specific pathways, these are neurons that fire when we engage in a particular action and observe others engaging in the same action. And the way that they discovered this was really neat. They were working with monkeys, and I believe they had just sort of electrodes that were monitoring individual neurons. And they found that the monkeys' brains would activate when they reached for food, the same way that they did when they saw the experimenter, a human, reach for food. So they were getting these mirror neurons activating not just to their own species, but to other species as well. So that shows some pretty interesting generalization and that they recognize the behaviors as the same. So that's really, really cool. And again, lots of cool research in this area as well. Okay, and I already said all of that. Cool. All right, so next we're going to start talking about different parts of the brain and how we study what is going on in different parts of the brain. So most of this is going to be talking about the limbic system. In earlier emotional research, everyone kind of assumed that all of our emotions originated within the limbic system, that everything came from the limbic system and it was sort of the be-all and end-all of emotions. But we've found a lot more lately that it's not necessarily as precise or refined as that. And we've already started to get this picture from our other chapters where we have, in order to experience something or in order to perceive something, we rarely activate a single area of the brain. It's usually a combination of different areas working together in particular patterns that produce certain perceptions. And it's the same with emotion. So yes, the limbic system is very important and it's going to be tied in with a lot of emotions, but we don't want to think of it in isolation. So even if we talk about, we know that the amygdala is involved in fear, it isn't that fear only comes from the amygdala, it's just that we have a really strong association. We know that the amygdala is active in certain patterns when we experience fear. We're not excluding the rest of the brain, we're just focusing on the part that we really understand as much as we can. But for our limbic system, we're going to be looking at sort of the area that's in that image there. So it's a whole network of structures that are arranged around our brainstem, sort of the upper part of our brainstem. And it's not just involved in emotion, it's also involved in things like motivation and learning and memory. So that's sort of leaning into that idea of it's not just for emotion and it's not exclusively for emotion. So it's going to be activated for other roles as well. And so all of these structures have multiple different functions that we can look at. Okay. So how do we actually study emotion? And when it gets to situations like this, where we're looking at a concept that is fairly vague in and of itself, we have to be kind of careful how we study things because we need to have a really clear operational definition. That's why I said most studies are going to state what definition of emotion they work with. They're going to tell you what they're looking at, and they're going to tell you what they're measuring as a stand-in for emotion. Because it's really, really hard to actually quantify feelings of emotion. So we might just measure some kind of physiological arousal. Is there activity in parts of the brain? Is there an increase in skin conductance? So we've talked about skin conductance before. That's just when we have electrodes on our skin and we'll see an increased transmit of electricity across our skin surface as we sweat more. So if we're experiencing higher levels of arousal, we tend to have more sweat. That's why our palms get sticky when we have to give a presentation or do something that makes us nervous. So we can actually measure that level of arousal by skin conductance. We could look at hormone levels. That would give us certain measures. We can look at, I guess we could go more of a psychology, pure psychology route and maybe get surveys asking people to report their feelings and maybe score the intensity. But all of these could be a measure of emotion, but none of them are going to encapsulate the whole thing. So just keep some of those limits in mind as we're working through this stuff. But most of the early studies looked at lesioning and stimulation in different parts of the brain, especially in non-human animals. So this would be working with non-human animal models. So lots of this work was done with, say, rats. Rats are probably one of the biggest just because they're easy to house and you can control lots of other factors that might be involved. But we have a lot of issues with that because, maybe you know that stimulating this part of a rat's brain produces aggressive behavior, but it's not possible to quantify what the rat is experiencing. They can't tell us how they're feeling. So we have to make some assumptions based on observed behavior, or based on cortisol levels that are circulating in the blood, or based on heart rate, or whatever we choose as a stand-in for actually having some kind of emotion that we can describe. So some of those drawbacks there. We also had some really interesting research sort of coming into the 1960s. So Robert Heath was one of those big names at the time, where he ended up co-opting patients who had electrodes in their brains for other purposes in order to learn what he could about emotion. So basically, they had, or he had patients with electrodes in their brains in attempts to treat things like epilepsy and sleep disorders, or even extreme pain that didn't respond to treatments. So they were using electrical stimulation to treat other things, which was how they were ethically able to do this, even though in the 60s, the ethics were maybe not as strict as they should be. But he was able to, in placing those electrodes, learn a little bit about what happened when different parts of the brain were stimulated. So if you think of a patient in one of those medical dramas where they're working in the brain and they're trying to find the specific part of the brain that's causing a problem, whether it's seizures or tremors or whatever, and you sometimes have to move the electrode around, supply a very, very small current to stimulate different areas until you find the place that fixes the problem. While you're doing that, if you document what happens when you activate other areas, then you can learn a little bit about what's going on and which parts of the brain might be associated with different emotions. So this is how they kind of got around the fact that you shouldn't put electrodes in people's brains to see what they feel. They were saying, since we're already there and there are already electrodes, let's make use of the information that we get access to. Okay, and so nowadays we don't see a whole lot of that. Most of the research, especially modern research, is going to lean into brain imaging, and this is a combination of increases in ethical structures and rules and everything. So it's a lot less likely to be able to do exploratory research like that. But also our technology has advanced to the point where we can get information that's just as useful and sometimes even more accurate using brain scanning techniques. So we can have highly detailed fMRIs or functional MRIs that tell us which part of the brain is active when we're doing something. Or we could look for PET scans or sort of any of the brain techniques that we've talked about that show activity. And we could start doing correlational studies where we have individuals in, say, an MRI machine, and we ask them to tell a story about something that made them happy or sad. And we can look at that differing activation depending on what emotion they're recalling. Again, we still have drawbacks where we're relying on people to basically have the same concept of what happy or sad is. And we do have to, again, keep in mind that our brains are not only going to light up in the areas involved in those emotions. If you're telling a story that made you feel an emotion, you're also going to be telling a story. So there's the motor part of speaking. So that part of your brain will be lighting up. You'll also have sort of other things. Maybe if you're talking about a time you went for a walk, maybe you have some activation in thinking about going for a walk, the action of walking. So we have a lot of background information that pops up in these studies. And I actually worked with someone, or I TA'd for someone who did a lot of brain imaging studies. And the way that they get around this is by conducting lots and lots of different trials and sort of subtracting out the background noise. So if they're specifically interested in what does your brain do differently when you're happy versus neutral, they'd get you to tell a story of when you are happy and then tell a story with similar elements that does not evoke happiness. So if you were talking about walking and walking in a place that made you happy, they would say, well, imagine walking to work, provided that work doesn't make you sad. And then they'd see, okay, well, this is the stuff that was consistent in stories about walking, and this is the stuff that's only lighting up when you're specifically happy about it. So they do some sort of fancy magic in the background to pull out different aspects. But if you've ever participated in a brain study, you might know that they take a very long time because they have to take lots and lots of control images and figure out what your baselines are. And each individual's brain is going to be different. So when we look at data, when we look at our sort of single image showing, look at the activation in this area, it's never going to be from a single trial. And And usually it's going to be averaged across, say, hundreds or even thousands of trials, and possibly even averaged across many individuals. So more technicalities to watch out for. Okay, so with all of that sort of background stuff out of the way, we can start talking about specific brain structures that we seem to know quite a bit about. So we can start off with our hypothalamus. And again, these aren't going to be new. We've already discussed a lot of these structures. But our hypothalamus, we had talked about having primary control over our autonomic nervous system. So our hypothalamus does a lot of our gut reactions to things. Our fight or flight response would be sort of triggered by our hypothalamus. So it produces general autonomic activation. So if we stimulate this part of our brain, if we have rats and you provide electrical stimulation, or if we were looking at humans with sort of activation applied to this area in the olden days, we would see general levels of autonomic activation. So it would trigger our fight or flight response. And when we could ask individuals about what they were feeling when this part of the brain was active, that'd usually be accompanied by sort of general feelings of fear, rage, and sometimes pleasure, depending on which part of the hypothalamus was stimulated. So it's not, again, it's more complicated than just this particular structure links to this particular emotion. We can actually have different parts of that structure sort of changing different kinds of emotions. So all of these are increased arousal, but some of them are due to fear, some are due to anger, and some are due to pleasure. So a couple of different approaches there. And this would have been nice to talk about when we were mentioning our different brain studies and where we tend to see activity based on which emotions that we're feeling. So this diagram here would look at, this is sort of condensing not just sort of across multiple trials per individual, this is actually looking at multiple studies. So they're looking at, so each square represents a single study for a particular emotion. And they looked at our sort of big five, the ones that people tend to agree are fairly universal: happiness, sadness, disgust, fear, and anger. Those are the ones that seem to be pretty consistent across all humans. And we do see that there is some clustering for each of these emotions to particular areas. There's also a lot of mess there. It's not as clean as we might like it. It's not like all fear only comes from one place. We do see it clustering on the area we're going to talk about next, where there's lots of activation around the amygdala, but it's not the only structure. So just more leaning into that idea that there's more going on. But since we mentioned the amygdala, we can actually talk about it. So this is another structure in our limbic system, and it's going to be located near the lateral ventricle of our temporal lobes. So I can actually hop to the next slide, and you can see it here, just sort of in that lower area. Move my mouse over to there. So if we go back to our brain imaging, and we're kind of thinking, okay, that was looking at the brain from the front. This image is looking at the brain from the side. That pink on the far side there would be where the amygdala would be. That's why we see a lot of pink there, which kind of makes sense because the amygdala is very strongly associated with fear and anxiety. And there's lots of research that's sort of leaned into this. And actually, the amygdala is one of the two most reliable associations that we know about. So this is one of the things that they're actually very certain about and that they've studied extensively. We'll talk about the other one in just a minute here. But they actually have a mutation that they've discovered. So if they have a mutation in the SLC6A4 allele, which is awful. But it's basically, it's a particular gene that's involved in serotonin production. And so if you have a mutation there, so saying that it's shorter probably just means that there's a piece that's been removed. So it means that you're not producing the protein that it's associated with correctly. That leads to reduced serotonin and increased fear and anxiety due to a hyper-responsive amygdala. So something's going on internally there where that particular mutation leads people to have an overactive amygdala. So lack of serotonin production is leading to that being sort of more active, and they are more fearful in general. We can also look at, again, back to our brain stimulation and damage with rats. If a rat has a lesion or damage to the amygdala, they don't seem to produce a fear response. So the text talks about rats who had lesioned amygdalas, who would approach sedated cats and interact with them, showing no fear of what should be a natural predator. So rats with a normally functioning amygdala would be cautious around a cat and would try and get as far away from it as possible. But these ones with damage to the amygdala would approach it, would interact with it. They even go on to say that some of the rats would bite the cats, and if the cat reacted, even to attack them, the rats would still hang around. It wasn't enough to get any kind of fear response from them. And so when we see issues in humans where they've had damage to both sides of their amygdala, they end up not showing traditional fear responses. But there is a caveat here. They don't show fear responses to external stimuli. So they aren't scared by horror movies or large predators or things like that. They don't have those sort of externally triggered fear responses. But looking at studies that expose people to, I believe it's carbon dioxide, which stimulates suffocation, in studies that expose us to that produces a fear response. And it produces a fear response whether you have functioning amygdalas or not. So even individuals with damage to the amygdala can show fear responses, but only to internal stimuli, not to anything external. So that gives us some evidence that there might be different pathways involved in interpreting internal versus external sources of fear, which is kind of cool. Okay. And again, the amygdala isn't just about fear. We know it best for fear, but it also ends up being activated with sexual excitement. It's involved in risk assessment. It helps us with decision making, and it activates when we're forming memories. So again, multiple things running through the same area. It's not just emotion, but we've very extensively studied it as it relates to fear and anxiety. Okay, so our next of our very reliable structures is to look at the insular cortex and basal ganglia. So again, we can hop back here. We can see our insula and our basal ganglia are highlighted sort of in that middle part of our brain. And these are very, very well studied, and again, very, very consistently studied and linked to feelings of disgust. So we've done lots and lots of studies that have shown these areas light up when we feel disgust. And it kind of makes sense. So disgust is typically related to things that, say, taste bad. And that's where the name comes from. So disgust means bad taste. So we do experience disgust to other things, but it comes primarily from something that sort of tastes bad and sets us off that way. But the insula, that's the cortical projection site for taste. That's where our taste senses are going to be sent for processing. So it kind of makes sense that that would be activated if we're experiencing disgust. And then our basal ganglia is involved in motor functions. So I just had a moment of like, lost my train of thought there. Okay. Sorry. So it's nearby, it's involved in motor functions, so it would be involved in our reaction, sort of that disgusted face, maybe pulling back from something, our standard activation in terms of disgust. So that kind of fits there. And there is some really interesting research. So people with Huntington's disease, as well as individuals with obsessive compulsive disorder, they both have abnormalities in the basal ganglia, and they both show consistent inability to recognize facial expressions of disgust. So in people who have lesser functioning basal ganglia, they have trouble identifying disgust on the faces of others. So something about that sort of interferes with their processing of that emotion, which is pretty darn cool. Okay, and then the last area that I wanted to talk about here is our anterior cingulate cortex, our AAC. And we're going to talk about this AAC quite a bit more as we kind of move forward. But just to get us oriented here, so this is a brain structure specifically known for a role in attention, decision-making, and impulse control. So that's how we've mainly studied it. So especially in terms of, if you think about it, decision-making and impulse control would definitely make you think about, say, gambling. And a lot of research on decision-making and impulse control does look at gambling. And in the scientific literature, gambling doesn't have to just be, going to a casino and playing cards or whatever. It can also be things like how likely are people to choose a risky decision over something that would be more safe, and how do the odds of a payout sort of affect those choices. And so I've worked with someone at the U of A, Dr. Spetch. She does gambling research, but with pigeons. She also does humans, so she can compare both of them to each other. But what she'll do is she sets up situations where pigeons have the option of choosing one of two doors. So the pigeons get a touch screen and they'll peck on the blue door or the yellow door. And one of the doors pulls from a pool where you have a high probability of sort of a small reward. And the other one pulls from a pool where you have a low probability of a very large reward. And we see how they weight the attractiveness of each of those. So maybe you're better off overall to get a consistent small reward than get sort of an inconsistent large reward. Maybe the payoff is much better to choose one door over the other. But if we have sort of differential functioning in this anterior cingulate cortex, we can have differential preference for things that are risky versus not risky. And this isn't just for gains. We also see some interesting research looking at losses. So if something is framed as maybe you start with $100 and you have the opportunity to either lose a small amount consistently or a sort of lesser probability of losing everything, then which one do you take? So you might have a preference for, if I go with the very, very risky option, I might have no withdrawal, but I might lose everything. But if I go the other way, I for sure lose something. So which one do I go with? And so we can start looking at patterns of deciding that way. So that's where a lot of the research is set up when we look at decision making. And we can see sort of consistencies in terms of behavior and risk taking with those sorts of choices. All right. Let me just see if I said everything I wanted to say here. Okay, so just for our general of what is going on here, I've made the point on the slide that it plays a major role in emotion. We haven't specified which emotion, and the reason for that is because it actually is a location where we combine emotion, attention, and information from the body, which kind of makes sense. So it's where we make sense of things and decide how to assign values or what we take from a situation, which would be why it plays such an important role in evaluating risks and making decisions. So it's the area where you're putting all that information together. So if we have problems integrating information, maybe if we have biases where we underweight risks and overweight rewards, we could see some shifts in our decision-making process. There are some studies that are less consistent. Some of them will link this to either sadness or happiness, depending on which study you look at. But again, lack of consistency, which we saw in our diagram with all of the squares on it, where lots of different studies can point to lots of different areas being active for lots of different things. Okay. And I think... Yeah, and I think that was it. So I wanted to end with sort of that cautionary note. We've kind of been introducing this as we've gone. I wasn't sure how well I'd integrate it during the lecture when I was setting up, but we're going to stress that idea that we don't want to assign emotions to particular brain structures. We need to understand that any particular emotion is going to activate an entire network. So even though we know that the amygdala is associated with fear, it isn't the only cause of fear, and it isn't the only thing active when we feel fear. So just to keep that distinction in your minds as we move forward and as we talk about more of these different areas. And also keep in mind that we have a lot of disagreement between studies. So depending on the methods that they're using, or what they're choosing to look at, or even how they're choosing to look at it, we can end up with a lot of conflicting research, which is why we tend to focus in on what's the consistent story that we get from the literature, but also here's some things that tend to pop up. So we'll make a note that, our ACC, is involved in decision making, but maybe also fear and maybe also sadness. So we have a lot of agreement on the decision making thing, less so on the fear and sadness. So when we're talking about things that way, that's why we're talking about it like that, because we don't have a solid 100% answer. We just have sort of this is our best interpretation given the state of the literature at this moment. And on that note, I will leave us here, and we will pick up with the rest of this chapter after our midterm. So we are good there.